is those molecules are oxidized. Right? All the hydrogens are removed, and the hydrogens go on to NAD plus or FADH to become NADH and FADH2. That's where the hydrogens go. Okay. Now, these are series of steps, right? You can look up those series of steps. There are multiple steps to be most energy efficient because those hydrogen atoms are where the energy is. Hydrogen atoms were part of a covalent bond. Covalent bonds were storage of energy. And then the CO2 we breathe out, right? The CO2 diffuses out of the cell. And then for us, it goes through our circulatory system and our respiratory system, and we breathe it out. Okay. Now, what we really talked about last time was one specific fate of those hydrogen atoms. So where do the hydrogen atoms go? Because what we really need to do is we need to regenerate the NAD plus and FADH so that the cell can continue glycolysis in the citric acid cycle. And remember, make a small amount of ATP. Still ATP, we're not going to turn it down, but there's just a small amount of ATP. Okay. So we need to continue that or those catabolic processes. And the only way a cell can continue that is if it gets rid of the hydrogens to go back to make more NAD plus, so they can do it over and over and over and over and over. So what is the fate, so to speak, here, of those hydrogens? And what we talked about was oxidative phosphorylation. That's really where the grunt work is in cellular respiration. Glycolysis and citric acid cycle are easy. This oxidative phosphorylation, remember, we have an electron transport chain. And chemiosmosis, that's how we define oxidative phosphorylation. And where did those hydrogen atoms go? Well, they went to oxygen, right? So if we look at this big picture, the hydrogens that make water at the very end of oxidative phosphorylation are the hydrogens that were once in your food. And a bunch of other stuff happened in between, right? Electrons, proton concentration gradients, and the synthesis of ATP, all dependent on the structure of membranes. If a cell and an organism utilizes this is what we call aerobic respiration. Because aerobic from oxygen, okay? Not all life utilizes aerobic respiration. There is diversity in metabolic needs across life, and bacteria have the most diverse. Okay, so I'm going to give you some other examples here. It doesn't have to be oxygen at the end of the electron transport chain. It could be other inorganic molecules. These could be phosphates, they could be sulfates, they could be nitrates. We can't do this. Animals, plants can't do this. Bacteria can, fungus can, okay? And when this is the case, this is referred to as anaerobic, if I can spell it correct, respiration. Same idea that we talked about before. The only difference is it's not oxygen. But you notice phosphorus, sulfur, and nitrogen also have high electronegativity. Right? So that's what's driving it. We just evolved to utilize oxygen. Okay? But some cells do not use oxidative phosphorylation. And there's a few reasons with why they may not use it. Some organisms don't have an electron transport chain. They just don't have one. And some organisms, if presented without any oxygen, they can go back and forth from oxidative phosphorylation to fermentation. I'm going to abbreviate electron transport chain here. So if the cell does not have an electron transport chain, or maybe it does, but there's no oxygen present, these types of organisms would use fermentation instead of oxidative phosphorylation. Us, as a species, we cannot use fermentation. There's only one cell type that can use fermentation and can only use it for very short periods of time. So really, us as a species, we are aerobic respirators. Okay, that is our main way to regenerate NAD+. However, fermentation is still very relevant to us. So where do you maybe know about fermentation outside of this metabolic world? Maybe you enjoy the benefits of fermentation. Wine, bread, cheese, yogurts, food. When we produce those, in my opinion, very delicious foods, we rely on the fermentation of microbes, bacteria, and yeast. And we have to understand the metabolic function of fermentation to do that food production successfully. The head, CEO, owner of Sam Adams has a PhD in microbiology. He understands the metabolic function of fermentation. Going into fermentation and food production, breweries, wineries, etc., as a microbiologist is a very lucrative career. You make a lot of money. Okay, but you have to understand this. Okay, So what is going on here? Well, they still need a way to regenerate NAD+, right? They broke down their organic molecules, but they can't break down their next lunch because there's no NAD+ around. So the final electron acceptors are organic molecules themselves. More specifically, the products of glycolysis. And I'm going to show you a picture here in a moment. There are multiple types of fermentations. And what I mean by that is multiple pathways that can occur. So where those hydrogens go, there's multiple ways or places those hydrogens can occur. Okay. Cheeses, yogurts, those organisms do lactic acid fermentation. They're producing the acids that give cheese, yogurts, those types of foods their flavor. Okay. And so here what you can see is you see glycolysis, right? Glucose to fermentation. I'm sorry, every time I hit my cursor button, it shifts the thing. So we see here glycolysis. And here's where we made those NADHs. But we have to remake the NAD plus or we can't keep doing glycolysis. So see what happens is the NADH donates the hydrogens to pyruvate. So pyruvate is actually a product of glycolysis, okay? And so in that process, it becomes lactate. You can have a very similar process, but instead of lactate being produced, you get carbon dioxide and ethanol. That carbon dioxide is what makes our bread rise and that alcohol we enjoy, right? These are not important to the organism. That yeast doesn't give two craps about that alcohol. It doesn't care about the CO2. It doesn't care about the lactate. What it cares about is the ability to regenerate NAD plus so it can go back here and continue glycolysis. And why is glycolysis important for an organism? Because it makes it ATP. There's no electron transport chain. There's no proton concentration gradients. So there's not a ton of ATP that's going to be made by organisms that use solely fermentation as the way to regenerate NAD+. In fact, there's no ATP made by fermentation. The ATP is made by glycolysis because we regenerate NAD+. This is what's important. Okay. And so if you are that microbiologist growing that yeast in the brewery, or if you have brewed at home, you have to give these yeast cells a ton more sugar than you would if you were giving them oxygen. So you have to close off the oxygen, so you have to seal them up in an anaerobic environment, and you have to give them more sugar, and they will switch to fermentation. So some organisms can switch back and forth, and we utilize that to make food. They don't want that, right? That's not their preferred environment. Their preferred environment is probably to do all of this that we talked about last week, but they can't because you closed off the oxygen. Okay? So they have no shift to this process, so they can just use fermentation. So it's understanding that organisms and cells vary in their metabolic functions. So questions on that. So this is really the big picture here, right? We're regenerating NAD+, and we can regenerate either with oxidative phosphorylation or with fermentation.
What is the pyruvate again?
Pyruvate is a product of glycolysis. So glucose, for example, if we're using glucose as the start, so glucose is a 6-carbon sugar. Pyruvate is a 3-carbon sugar. So it's a slightly oxidized form of glucose. I said that I would talk a little bit about drugs and poisons. So here is this figure that accompanies this discussion. So we know that carbon monoxide is not good for us and carbon monoxide will kill us. So how does carbon monoxide work? Well, carbon monoxide blocks this last transfer of electrons to oxygen. So if it blocks this continuation of electron transport, it's also blocking the pumping of hydrogen, right? Because the electron transport chain is driving the concentration gradient. And then if you don't do that, you don't get to make any ATP. So your cells starve, basically, from carbon monoxide poisoning. Rotenone is an anti-parasitic, so we use it in like insecticides like ticks and fleas and other insects that cause diseases, and it stops the electron transfer earlier on in the pathway. Oligomycin is an antifungal drug, and what it does is blocks the hydrogen protons from being able to diffuse through ATP synthase, thus again starving the cells from ATP. And this is an interesting one. So DNP, which stands for dinitral phosphate or something pyruvate, something like that. Brain just blanked. I usually have it memorized and just blanked on me right now. It actually makes the membrane leaky for protons. Remember, the whole reason this works is a membrane dams up protons, like the Hoover Dam, right? We're damming them up and forcing them to go through ATP synthase. Well, if they can just diffuse from high to low right through the membrane, they don't need to use ATP synthase. And if they don't use ATP synthase, the cell's not making ATP. DNP was common in diet pills in the 90s. And the FDA pulled them for very good reasons because individuals that consumed those diet pills lost all weights, not just the weight they wanted. These very shady health websites, at least as of a couple years ago. I haven't looked in a couple years, but there are places claiming to still sell them, claiming to have backstock of those pills from the 90s. One, they're probably lying. They're probably not really those pills anyway, and they're just taking your money. But two, it's will kill you. So the FDA pulled them for a very good reason. Okay, so a couple questions in the chat. So Kaya says, can some bacteria use either fermentation or oxidative phosphorylation depending on its environment? Yes, and yeast does that. So yeast is the one that we use mainly for brewing and beer, or brewing and bread production. We use yeast. You probably have a jar of yeast in your fridge if you bake in those types of things. So they can switch. And so you have to switch their environment on purpose to get them to switch to use fermentation. Absolutely. And Miriam, I'm not sure what 3 you mean. I'm probably going to assume you mean these 3.
Can you just, I couldn't write so fast. Can you repeat what the first one blocks
Absolutely.
the hydrogen and what are the other 2 chemicals do?
So when we make the NADH in glycolysis in the citric acid cycle, we cannot keep doing that unless we regenerate the NAD+, meaning the NADH has to give their electron somewhere. So they can give it to one of 3 places. They can give it to oxygen via an electron transport chain in chemiosmosis, and that would be called aerobic respiration. They can give it to another highly electronegative molecule like phosphates or sulfur or nitrogens, and that would be called anaerobic respiration. Or they can give it to organic molecules, usually products of glycolysis themselves, and we call that fermentation.
Oh, I'm sorry. I meant the 3 chemicals.
Oh, okay. Sorry. So they're just, so carbon monoxide and cyanide are poisons to humans, right? And so they're blocking electron transport chain right here. Rotenone is an anti-parasitic, it's an insecticide basically, and it blocks electron transport chain up here. So this yellow line is the electron transport chain. Oligomycin is an antifungal. and it blocks the diffusion of protons through ATP synthase.
So your body can still produce energy without the electron transport chain, right?
Your body cannot. Nope, you would die.
You have to have the. I thought there was 2 other ways we can.
Not us. Not us. Life as a whole can utilize any of those combinations, but us as an individual, we only use aerobic respiration. Muscle cells can use lactic acid fermentation for very short periods of time, but as soon as your muscles are done with that strenuous activity that you forced on them, they switch back to aerobic respiration. So those are 3 different possibilities for life as a whole. But any one type of life usually can only utilize 1, maybe 2 of those methods. Okay, so that's cellular respiration. So we're going to take a little breather here. First, I do. First, we'll do a little poll. So just a few questions to see if you just have the basic foundations down. So go ahead and take a minute or 2. There are 3 questions in this poll. And then we are going to do the activity, the lecture worksheet. So if you haven't opened that yet, from Canvas chapter 7. And just about 30 seconds left for the poll. Okay, I'm going to close the poll and share these with you. So in glycolysis in the citric acid cycle, the carbon atoms that were once in glucose leave as carbon dioxide. The carbon atoms, irrelevant. We breathe them out as carbon dioxide. What's important is what happens to all of those hydrogens. Oxygen is the final electron acceptor. And to go back to Preston's question is, you know what happens. I wouldn't test this. I do not recommend trying this at home, but you all know what happens when you stop breathing. You die. So pretty good evidence that we can only use aerobic respiration as a species. And number 3, The proton concentration gradient is the most direct source of ATP production and oxidative phosphorylation. You go back one further step, it would be the transfer of electrons. And if you go back one more step, you could say NADH. And then if you go one more step, you could say the food that you consumed. Okay. Any questions on those or anything for cellular respiration before we sum up with the worksheet for today? Well, for cellular respiration for today. We'll go into photosynthesis next. Ashley.
So the only reason on number 3 is I chose oxygen instead of the proton gradient because I was in between those, is that I remember you mentioning rigor mortis and how something to do, I have to look at my notes, but it's something to do with the oxygen and ATP. So does oxygen not play a role?
Oh, oxygen is important without oxygen, but oxygen is not the most direct source. So the most direct source is the proton concentration gradients. Okay.
Okay.
When I was talking about rigor mortis, I was using the example of the lack of oxygen, but that's not, rigor mortis happens when everybody dies, regardless of whether you die from the lack of oxygen. So rigor mortis is the immediate paralysis of all cells in the body due to the lack of ATP, and that happens death regardless.
Okay, got it. Thank you.
Yeah. All right, so if you haven't yet pulled up the worksheet from chapter 7, please do. I'm going to share my screen so that you at least have it. And I'm going to give about 10 minutes for you all to just work on this individually. Feel free to help each other out using the chat or just talk out loud if you so choose. And then at the end of that 10 minutes, we'll go over these together, or I'll just summarize any that you have a particular question on. Okay. So I have 1:16 on my clock. So 1:26 will sort of come back and just, summarize whatever you need from this worksheet. And I'm just going to stop sharing for a moment so that I can set things up for photosynthesis, and then I'll reshare it. So if you're looking at my screen, I'll be right back with that. And I have about 4 minutes left on my countdown. So let's see some of your thoughts for the answer to the first one. So whether you just want to say it out loud or put it in the chat. For every molecule of glucose, how many molecules in total of electron carriers can we make? Any thoughts? Anyone at all?
12?
12, absolutely. There's 12 H's in glucose, so we can make 12 electron carriers. For every hydrogen there is, that's how many get pulled onto an electron carrier. It doesn't matter if it's NAD, doesn't matter if it's FADH, 12 is the total. So nice job. What about number 2? What do we think the answer is to number 2?
Can it be two things?
No.
Well, then I'm going with carbon dioxide.
Okay. What is the oxygen used for in cellular respiration? What's the role of oxygen? I told you that on Thursday, right? This is our top thing, to know what the role of oxygen is by the end of this chapter. So what's the role of oxygen? Okay, oxygen is the final electron acceptor. Okay, so it's important to differentiate between oxygen as we breathe it in and the oxygen in organic molecules, right? So this is how scientists traced all of these things, right? So here, that oxygen would become water. Okay, so I don't know what the other answer choice that Preston had or anyone else had. So here we're talking about the oxygen in the glucose, and that absolutely becomes CO2. So nice job.
Wait, I'm sorry.
What?
The oxygen in the glucose becomes CO2?
Yes.
Okay.
Yes.
Yeah.
My brain failed me when I first read the question, and so I just went with my line of questioning to you because you need to know that anyway. When I reread the question, I was reading it as the other oxygen, so that's why I paused and was questioning you all, but you all got it right. So it was my brain that failed.
So what was the thing about the water?
Carbon dioxide is correct. Carbon dioxide is correct. Yeah. Okay. So what do glycolysis and the citric acid cycle share? What do they have in common? What happens in both of those?
They both make ATP.
Okay, you could say that. They both make ATP. They both make NADH. Lindsay says that. They're both catabolic. They're both oxidative, right? Any of those answers. Okay. So what about this pH difference? If you were to continue that red line at the point of the arrow, how would you continue it? Would you continue it straight? Would you go up? Would you go down? Would you go some other? What do you think?
It would decrease the protons in the inner membrane space.
Okay, it would do that. So on the y-axis here is the pH difference.
So that wouldn't it increase because the ETC is still spewing those H plus atoms into the intermembrane space?
Right. So what happens is because ATP synthase isn't functioning, you're not getting the diffusion of protons down their concentration gradient. So they're continued to be pumped. So the pH difference is going to go up because you're continuing to pump H pluses across the membrane. So you're continuing to make a gradient. And pH is a measurement of H plus, right? So the pH is going to be a lot different on the inner side than on the outer side of that membrane, okay? Now, over time, eventually it would taper out and the electron transport chain would stop pumping. It would receive that feedback. but immediately it's going to go up because you're sequestering H pluses on one side and not on the other, and that's a difference in pH.
Oh, is the hydrogen pump not controlled by the process of ATP synthesis?
Not by the ATP synthase itself. So it would eventually receive the feedback, because if you're sequestering all the H pluses on one side, then there's no H pluses coming back down, and then there's no more to pump, right? So eventually feedback in the cell would stop pumping, but immediately they're continuing to pump. And so they're not directly reliant on ATP synthase in that sense anyway.
But just to clarify, so the protons they're still continue to be pumped inside the mitochondrial membrane, correct?
Absolutely.
So then wouldn't that make it more acidic if you're having more protons come in?
Yeah, absolutely. But this is the pH difference.
Oh, okay.
This is pH difference.
Okay, thank you.
Yeah. So one of the things that I will continue to remind you all of, because I just see it, it's common, is always make sure you know what the graph is showing. Don't infer anything else. So it says pH difference. So that's the little catch there. Okay. All right. So keeping everything in mind that we just talked about, we're going to talk about photosynthesis. Okay. Photosynthesis is going to be the same pieces. We're going to talk about electron transport chains. We're going to talk about proton concentration gradients. We're going to talk about ATP synthase, and we're going to talk about membranes. The puzzle pieces go together in a different way for a different purpose. Okay. So photosynthesis. Let me get some space here to give you some initial notes. So photosynthesis, from the name alone, you know, light's involved, and we're going to make something. Okay, so photosynthesis is the conversion of light energy. So a new type of energy that we haven't talked about. Well, you watched the pre-class video, but we haven't talked about it, right? Conversion of light energy to the chemical energy of food. So this is the production of food. I mentioned last time that we are consumers. Oops. So these organisms are called producers. And the formal word for that is autotroph. So means body or form, cell feed. So these are cell feed. Okay. Examples of producers, plants, bacteria are photosynthetic. And what structure allows for these organisms to do this are light absorbing pigments. There are several light absorbing pigments, but the main photosynthetic ones are chlorophyll. This process is also an anabolic process, right? Synthesis of things require energy. So energy is on our left-hand side of the equation. That energy is in the form of sunlight. And so this may just seem like the equation of cellular respiration backwards. And it is, and that's important. We'll talk about that in a moment. But it's the process isn't backwards. Okay. But what you also can see here is that we still are talking about movements of hydrogen atoms. However, in this case, carbon dioxide is reduced. So carbon dioxide gets hydrogens added onto it to become an organic molecule, and water is oxidized. Okay? And again, this is our broad overarching equation. It's not super balanced, it's not super detailed. It's looking at it as a whole. And remember, this is any organic molecule, not just glucose. Kelsey, go ahead whenever you're ready.
Did you mean to put C6H12O2 or
Nope.
O6?
Nope. Thank you. Okay. So same idea that we talked about with cellular respiration. We're going to follow the hydrogens. We're going to look at membranes. We're going to see conversions of energy, and we're going to make, in this case, food, right? Okay. We're going to use plants here as this example. Not all cells in a plant can undergo photosynthesis. But every cell in a plant needs organic molecules, right? Every single cell needs food so that they can turn that food into ATP via cellular respiration so they can do cell work. Photosynthesis and cellular respiration are not mutually exclusive. And I'm going to repeat that because I will ask you and someone will still miss it on the exam. Cellular respiration and photosynthesis are not mutually exclusive. Plants, algae, and photosynthetic bacteria do both. Photosynthesis is the production of food. Those cells then transfer the food to all other cells in that organism so that they can turn the food into ATP via cellular respiration. Plants do both. It's not either or, okay? Because only the leaf cells in a plant, like the trees that you see outside or the house plants you have, only the leaf cells are doing it. Once they have the food, they use the food in cellular respiration. Okay, we're going to come back to this picture in a moment, but what I want you to see here is the structure of the leaf. Well, it'd be my drawing, so we'll focus on this nicer drawing here for a moment. So leaves have three layers. They have an outer epidermis on the top, an outer epidermis layer on the bottom, and then they have all these cells in the middle. This is referred to as the mesophyll. So that's the tissue, the interior tissue. It's the mesophyll cells that contain chloroplasts. Okay, so here's a mesophyll cell right here, and then you see many chloroplasts. We're going to be zooming in on a chloroplast. We've already looked at this structure. We have an outer membrane. We have an inner membrane, and we have these little stacks, these little green discs. These discs are called thylakoids. And in the thylakoid membrane is where the chlorophyll is. What contains. Okay, so the membrane contains the chlorophyll. So these are all the levels we're zoomed in on, right? So I will repeat that. In the leaf, the middle tissue layer is called the mesophyll tissue layer. Those cells contain the organelle chloroplast. In the thylakoid membrane within the chloroplast is the chlorophyll with the pigment that we'll be talking about. Psst, there's a lot of membranes here, right? Every disc is membranes. Psst, lots of membranes. Okay? Because it's going to be the same thing as cellular respiration. A little bit different, right? Some different nuances, but the same basic premise. Now, in the pre-class video, you learned about light, right? Light energy is part of the electromagnetic spectrum. It is a particle moving in waves. The shorter the distance between the waves, the wavelength, the more potential energy, right? And then there's really damaging electromagnetic radiation, which we want to avoid at most costs. And then there's pretty safe radiation, right? Our cells are not going to be harmed by radiation on this side of the spectrum. And then there's this little visible light spectrum, which can be broken down into colors, each with a respective wavelength. And here are some pictures of what those light absorbing pigments look like. Okay, so you can just see them. And you see very similar to those cytochromes we talked about, there is a metal binding center. Okay? So this is going to be relevant to all the electrons we're going to be talking about, right? Because the electrons are attracted to those positive centers. Each light absorbing pigment only absorbs light at a certain wavelength. And most of our chlorophylls have an absorption over here in the blue-purple range and the secondary peak over here in the red-orange. So if you have grow lights in your house, they have red spectrums and blue spectrums, or you can turn them both on because that's the color of light that they're using. They do not use green. Sorry, I thought I had a little picture of it. I don't. They do not use green. Green is reflected. That's why my plant, which you can't see in the camera, my plant right here is green. Your plants are green. Most plants are green because they evolved from the same ancestor. Okay. Now there is variation of that. We have berries, red peppers, eggplants. We'll talk about that towards the end of today or maybe on Thursday, what those pigments do. Okay. They're not photosynthetic pigments though. All right. So we're going to make a drawing just like we did for cellular respiration. So we're zoomed in. Okay. So here's my amazing leaf cell. Okay. And in the middle of that, right, is the mesophyll. So this is a mesophyll cell. And that little green dot is the chloroplast. Right, so this little green dot is what we're zooming in on, and that's the chloroplast. Okay. And so what we have here is the outer membrane and the inner membrane. So technically those are both double. I just failed to make them as double membranes. Okay. And this is the thylakoid membrane. Okay. So we're in a single thylakoid inside a chloroplast inside a cell. Okay. So we can continue this, right? The thylakoid is like a disc, so this thylakoid would be enclosed, right? Okay, so we'll talk about that in a moment here. This fluid is called the stroma, right? So there's a fluid, oops, sorry. This is the fluid right here, so we'll come back to that too. But right now we're going to be zoomed in on this thylakoid membrane, okay? Photosynthesis can be broken up into two stages, right? These main stages. First one are very conveniently called the light reactions. Cell is going to turn light into ATP. We need ATP to do cell work, so we got to convert the light to the cell energy. So that's one thing that's going to happen here. We're also going to split water. For those of you that are budding botanists or that just want to keep a plant alive in your house, there are two principles to keeping plants happy: light and water. They go hand in hand. If you give them too much water and not enough light, or vice versa, they're not going to grow. And this is why. Okay. Now, water here is going to produce oxygen. Awesome. We need that oxygen. Thank you, plants. Okay. But the plant doesn't care about this oxygen. You put this in a more appropriate way. Here we go. We're going to take those hydrogens from water, just like we took the hydrogens from food, and we're going to put them on an electron carrier. In this case, it's NADP. NADP, FADH, NAD, who cares? They're all the same concept, okay? And all of this is going to take place in that thylakoid. So that's what we're going to talk about. How does light become ATP, and where do the electrons go? Okay, so let me zoom out here so I make sure I have enough room. The first thing we're going to draw are these larger complexes called photosystems. Okay, so these are photosystems. And then in between the photosystems is an electron transport chain. Okay. And then if we sort of zoom out here, remember that our thylakoid's a disc. So the inside of that disc is going to be relevant. Okay. And you could probably guess, if we have an electron transport chain and you know we're making ATP, then we also have an ATP synthase. Okay, so you know some of this already. Photosystems are really the only new thing that's happening here. I forget which colors I used last week for electrons and protons. If that kind of stuff helps you, use the same colors you used Thursday. I don't remember the colors I used. Okay. So I'm just going to pick new colors. Let's talk about the sunlight first. Sunlight, well, not purple sunlight. Sunlight is hitting the leaf. The sun's basking in the sunlight. The chlorophyll are going to absorb it, right? So this is light. The photosystem is a complex of chlorophyll and proteins. We care about the chlorophyll, right? So I'm going to draw some little chlorophyll in here. Green, because that's how we see them. Okay? Any pigment we see is the reflection of light, not the absorbance of light. Okay. The chlorophyll absorbs the light energy. What that does, it bumps the electrons in the chlorophyll to an outer shell. That gave them more potential energy. That bump in potential energy is short-lived. As the electron falls back down, the energy is transferred to another chlorophyll. And it's just chaotic energy being transferred all around. Disorganized. There is a special pigment in each of these. I'm not gonna stress about the details, but excuse me, these are considered accessory pigments. Sometimes they have the P760, P800 names. Great if you want to learn about that. Think big picture for me. These are going to be called primary electron acceptors. That's what we're going to call them. They now organize the energy. And what they do is, hopefully you guessed it, they send that electron through the electron transport chain. So this is the electrons. Okay. So this is a linear pathway. So if you do read a book about linear versus cyclical, I'm talking really more about this linear pathway. Okay. So from the first photosystem through the electron transport chain to the second photosystem. And then we'll talk about the final electron acceptor in a moment. So same thing we talked about. Electrons go through an electron transport chain and they make their way to a final electron acceptor. Okay. When you think electron transport chain, just as we did with cellular respiration, pooling of electrons through those molecules means they're gaining energy. And what do the cytochromes that make up that electron transport chain do with that energy? What comes next? Miriam?
They expel hydrogen.
Absolutely. Right? So hopefully you gave yourself more room on your drawing than I did. That's why I still wish I had a big board here. Okay, so H pluses are going to accumulate inside the thylakoid. We've trapped them.
Isn't the is the thylakoid the inside then, or is it the? Can you show which part it is? I guess.
Yep, yep. Sorry, I wanted to make sure I finished that and didn't write a weird word. Okay, so they're pumping them into the thylakoid. And in a second here, I will zoom out to show you what the thylakoid looks like. Okay. So nothing different here than cellular respiration, except the energy came from a different source. The energy came from light. And you probably can guess that those H pluses are then going to diffuse from high to low through ATP synthase. And that energy will take A, D, P, and P and join them together to make ATP. Right? So we have electron transport chain and chemiosmosis, same thing we did before. Okay, I'll give a minute and then I'll zoom out, Kaya. Okay. Oh, sorry. I was trying to type in the chat and my thing thought I was doing something else. So if we zoom out, what is happening here in the light reactions is very similar to what happened in oxidative phosphorylation, right? We made ATP through oxidation reduction reactions, through an electron transport chain, and through chemiosmosis. Now, if we look back here, right, each of those thylakoids is like this little disc, and so the membrane is circular. So what's happening is we're trapping the H pluses in the disc, because remember, they can't go through the membrane directly. They need that ATP synthase. And what you just drew is happening in hundred and thousandth fold in every thylakoid membrane. And there's hundreds or thousands of thylakoids inside a single chloroplast. And there could be hundreds of chloroplasts inside a single cell. So all this is happening in abundance at the same time. Okay. Now, in cellular respiration, we called that whole process oxidative phosphorylation, because we are phosphorylating a DP to make a TP. Same thing here, except it has a unique name, which is photophosphorylation. Photophosphorylation is the process by which cells use light energy, an electron transport chain, and chemiosmosis to make ATP.
Did you say that last sentence again?
Oh, go ahead. Who was that?
About the photophosphorylation?
Uh-huh. What about it? I'm sorry, Jack.
Just the last sentence you said about it, how it produces ATP.
Yeah, so photophosphorylation is the process by which cells take light energy, right, an electron transport chain, and chemiosmosis to make ATP. So everything you just wrote. So everything you just wrote, we can also call photophosphorylation. Now, calcium is the nice seed to the segue. So there is questions here. Where did the H pluses come from? Where did the electrons come from? In cellular respiration, we knew they were coming from food because this whole process happened last. Enter water. Water serves two roles here. Well, really one main role, okay? Water is the electron source. All of these electrons, if they get shuttled through the electron transport chain, the chlorophyll is going to run out of electrons. So we have to replenish the electrons, okay? And that's what water does. Try to find a color I didn't really use yet, which I don't have very many color choices in in Outlook here. So we'll just use black, okay? So water is what's adding the electrons. That's why water and sunlight go together. Keep your plants happy. If you give them lots of sun, they need equivalent amounts of water. And the final electron acceptor, the electrons go on to NADP to make NADPH. Well, actually, technically NADP is the final electron acceptor. Okay, this is important. because this is only step 1, right? We got to move on to step 2. So the electrons go from water through all of this process and go on to NADP to make NADPH. And technically, sorry, if we're being technical here, which we should be, right? Water is actually in the stroma. Plus NADPH will make the food. And that's what we'll talk about Thursday after your exam. What I hope that you're starting to see as more and more of this is entering your brain and your brain is processing this, it's all the same pieces. In cellular respiration, the oxidation happens first, and then the electron transport chain and chemiosmosis. In photosynthesis, the reduction happens first, right? We're reducing, well, actually, we're oxidizing water, sorry. The light happens first, right? Electron transport chain and chemiosmosis happens first. Then that ATP, then that NADPH is going to take teeny little carbon dioxide and build it up. Anabolic reaction, reduction of carbon to make the food. Same pieces, different order for different purposes. Any last musings, thoughts, confusion?
Would you be able to make it a little bit smaller so I can see the whole thing? Please.
Is that good enough? It's like so small on my screen, but if you have a big screen, I guess it's not too small. Okay. Now, remember for your exam is on chapters 5 and 6. No cellular respiration, no photosynthesis. However, it's all the same stuff.
Okay.